Hi there, this is FemmeSciComm's fourth vlog. I post videos and written posts on FemmeSciComm.com weekly about published women's health research studies. I made FemmeSciComm so women can learn more about their reproductive disorders and their whole bodies through the reproductive system. Remember, up until relatively recently, research studies were mostly done on men. So the effect of hormones on the female reproductive system and female bodies in general is still deepening. This week's post focuses on one of those hormones, progesterone. So progesterone, along with its progesterone-like compounds, progestins, through the progesterone receptor, has been implicated in many reproductive disorders such as breast cancer, endometrial cancer, endometriosis, and fibroids. This week's paper, The Actions of Progesterone in Fibroids, parallel its actions in breast cancer. It's still October and it's Breast Cancer Awareness Month. So let's talk more about the functional aspects of progesterone. In this paper that was recently published in Oncogene, progesterone receptor integrates the effects of mutated MED12 and altered DNA methylation to stimulate RANKL expression and stem cell proliferation in uterine leiomyoma. The first author is Shimong Liu, and senior author is Sardar E. Bulun, both from Northwestern University Feinberg School of Medicine. So let's get into it. This study aims to determine the mechanism of progesterone receptor, PR, in promoting fibroid tumor growth. First, a little bit of background. What is progesterone? Progesterone is a steroid hormone that is essential for normal female reproductive function. As I mentioned previously, progesterone, through PR, acts out functions important throughout the reproductive tract and beyond. One function PR has is through its genomic functions. It is a transcription factor that can bind to certain regions of DNA and regulate gene expression. Briefly, transcription factors are proteins involved in reading and regulating genes in DNA. They are partly responsible for tissue-specific regulation. Think about it. Each of our cells have the same DNA in them. How do our eyeball cells know what to do versus our stomach cells? Different genes are turned on in different tissues in order to do its programmed function. Transcription factors can turn on these genes. They're important players in the field of epigenetics. When you hear the saying nature versus nurture, nature is genetics and nurture is epigenetics. We'll talk a little bit more about aspects of epigenetics a little later on. So one gene PR is hypothesized to regulate is RANKL. This gene name is short for receptor activator of nuclear factor kappa B ligand. A couple studies found a positive correlation of progesterone and RANKL levels. And previous studies from this research group found that blocking the RANKL pathway stops fibroid tumor growth. With this information, they hypothesized that PR through RANKL progresses fibroid development. The researchers first compare RANKL mRNA and protein expression levels in myometrium compared to matched adjacent fibroid tissue. Both mRNA and protein levels of RANKL are much higher in fibroid tissue. When they stain slices of these tissues with RANKL antibody, they find that a higher percentage of cells in fibroid tissue versus normal myometrium have RANKL present. Next, using cell surface markers, this research group previously identified as distinguishing important cell populations in fibroid tissue, they sort out the 3 different cell populations, treating the tissue with a vehicle control and with the actual RANK ligand. With the RANK ligand, they observed an increase only in the stem cell population. Via a proliferation assay using CCK8, they determined the mechanism of this increase was due to stimulation of stem cell proliferation. Because their overall hypothesis postulates that RANKL is being driven by PR, they treat normal myometrial tissue and fibroid tissue with R5020, which is a progesterone agonist, or you can call it an activator, and RU486, a progesterone antagonist, or we can call it an inhibitor. RANKL mRNA increases with the progesterone activator and decreases with the progesterone inhibitor, indicating RANKL is being driven or regulated by PR. Via ChIP-seq, ChIP-qPCR, and 3C-qPCR, the researchers pinpointed 2 areas of DNA they established that PR binds to. The first region is a relatively nearby enhancer. In epigenetics and genetics, enhancer regions are short regions of DNA that can be bound by proteins to turn on a usually close-by gene. They find that PR binding is increased at this region only in fibroid tissue. They also find that this increase in PR binding is positively correlated with the increase in RANKL mRNA expression. The second region they found that PR binds to is a promoter region, which is a region of DNA that is proximal or close to the gene being encoded. PR binding is also increased at this region only in fibroid tissue compared to normal myometrium. So they suggest only in fibroids, PR binds to these regions, thus increasing expression of RANKL mRNA. So I previously began talking a little bit about aspects of epigenetics. Epigenetics is the study of changes in biological mechanisms caused by modifying gene expression rather than changing DNA itself. One way of modifying gene expression is through DNA methylation. Picture a hoarder's home. Lots of boxes inhibit or stop you from reaching that person physically. Here, boxes are methylation. High methylation means a specific protein, or U, can't get to the gene, the hoarder, they want to regulate. Low or no methylation means the protein, U, can get to the gene, the hoarder, you want to regulate. Via methylcap-seq and methylcap-qPCR, the researchers determined there is high methylation at the RANKL enhancer region in normal myometrium. However, in adjacent fibroid tissue, the methylation level is much lower. Instead of a hoarder, you have the beginnings of a pack rat. To determine if this methylation indeed regulates RANKL transcription, they treat the tissues, both myometrium and fibroids, with a DNA methylation inhibitor, 5-AZA. Think of 5-AZA as the movers who clean up the hoarder's house. With this treatment, they observed an increase in RANKL mRNA as well as an increase in PR binding to this region. Finally, they look at this mechanism in a subset of fibroids that have a specific mutation in their MED12 gene. To determine this, they genotype the fibroid tissue to confirm the mutation that they call G44D. They find that RANKL is highly upregulated in the mutant fibroids. Also, they found that not only does PR bind to the RANKL enhancer region, but the mutant MED12 protein does as well. They find that MED12 binding at this region correlates with the high expression of RANKL mRNA, as well as high PR binding. To add more evidence of this MED12 PR interaction, they utilize immunoprecipitation followed by immunoblot to pull down the MED12 protein and blot with PR, discovering the interaction of MED12 and PR in fibroids with the MED12 mutation. So what does this all mean? This study details the mechanism of the progesterone receptor in the development of uterine fibroids. The mechanism that they detail incidentally confirms that the use of birth control, such as Lupron, a GnRH agonist, and Ella, which is a selective progesterone modulator, may help in stopping the growth or the symptoms of uterine fibroids, if progesterone is really initiating stem cell proliferation in the fibroid. However, the use of birth control may not completely stop the growth of the uterine fibroids or the symptoms of it. The researchers also pinpoint the importance of DNA methylation in uterine fibroids. It may be interesting to see if DNA methylation inhibitors like 5-AZA can be used as a therapeutic option for uterine fibroids in addition to birth control. Moreover, it seems like having the MED12 mutation that they describe leads to more enriched binding of RANKL by progesterone receptor. It would be interesting to find out if the women who have this specific mutation have larger or even more painful fibroids. This mechanistic study has several clinical implications and possible future directions, but don't forget the caveats. For example, a huge reminder that I have been talking about since the first video, correlation studies do not determine causation. Just because they observe high MED12 enrichment at the RANKL enhancer doesn't mean it is what causes high RANKL mRNA expression. In the same respect, just because there is higher enrichment of PR at the enhancer region doesn't mean that it recruits MED12 to that same region. Next, in their last figure, the researchers go over MED12 mutations. But what is the mechanism of gaining this mutation? What percentage of all fibroids have this mutation they describe? And I've mentioned this before, but what are the clinical implications of this mutation and others similar to it? Also, it's a caveat, but probably a future direction for this research group. All experiments are done on human tissue and cells. It would be interesting to look at mouse models of uterine fibroids and determine if the use of a DNA methylation inhibitor will affect tumor growth and pathogenesis. Overall, this study pinpoints novel therapeutic targets, RANKL and DNA methylation, for the treatment of uterine fibroids and suggests the importance of personalized therapeutic strategies for women with fibroids that harbor specific mutations. Thank you for reading, listening, watching my assessment of this paper on uterine fibroids. Keep informed, stay interested, and keep doing great women's health research. If you're interested in this content, please make sure to subscribe to my page. You can also follow me on Facebook, Twitter, Instagram, and Pinterest, and check out my website, FemmeSciComm.com. Thank you. Bye.